My name is Peter Mitchell. It is the year 1961. I'm going to share with you my hypothesis about ATP synthase. I explained how the energy stored in the proton electrochemical gradient is used to synthesize ATP. Here's an informative film about the process.
Let's view the structure of a plant cell.
This is a 3D representation of a plant cell. The plastic rim represents the cell wall. The blue background represents the cell membrane. The pink ball represents the nucleus, and the yellow dots represent the nuclear pores. If you were to open up the nucleus, you would see a nucleolus. The paper clips represent the cytoskeleton. This represents the mitochondria. This represents the vacuole. This represents the lysosome. This represents the Golgi apparatus. This represents the rough ER. This represents the smooth ER. And this is the chloroplast, which we will now learn more about.
Now let's take a deeper look at ATP synthase. But before we do that, we will share some background information.
ATP services a multitude of functions in the cell, and without it, the cell would be a mess. ATP is like the food source for the cell. Chloroplasts are only found in eukaryotic plant cells. They contain chlorophyll, which gives plants their green color.
ATP synthase allows for the production of ATP, which in turn converts energy from the sun into usable energy for the cell. Without it, the cell cannot survive.
This is a 3D model of ATP synthase. It occurs in the membrane of a chloroplast. Evan's arms represent the chloroplast membrane, and above his arms there's a high concentration of hydrogen cations.
ATP synthase is composed of two major parts, F sub 0 and F sub 1. F sub 0 forms a proton channel in the inner membrane, allowing for protons to flow through. F sub 1 is where ADP and PI, or inorganic phosphate, join to form ATP. This process occurs once three charged hydrogen ions travel across the gradient and into the intracellular matrix. Once three have passed through, there is enough energy for ADP and PI to join and create ATP. The process completes when the gradient disappears or when there is an equal number of protons on each side of the membrane. In this case, however, a gradient is always maintained. Rachel will now show how the process occurs. Now there's enough energy for inorganic phosphate to come and join the ADP. And now we have ATP, which is used for energy in the cell.
What makes our presentation unique?
We chose everyday objects to represent the cell and allow a more comprehensible way of learning the complex process.